So I'm gonna switch kind of gears now to a very more, I would say, more empirical presentation from the point of view of a marine ecologist. I'm a trained originally as an experimental marine ecologist, and that has been exploring over the past few years different approaches that help us understand coastal ecosystems in general and issues of sustainability, issues of conservation, and in which way we can help to provide information that can stop the madness that we are in at this point in the state of affairs of coastal ecosystems, which is not very good, as you might suspect. So I'm not going to show you any really of mathematical approaches in the talk, and some of it is going to be a little bit basic for some people. So sorry if I offend someone by going through some things that are probably very basic. And as we've seen in the more ecological presentations, there is, like someone said, that no species lives alone. And now that introduction that I had well prepared is kind of challenged by the previous presentation of Pablo, where you can sustain some systems apparently almost by themselves. But in general, most living things in nature depend on other organisms and established relationships that allow them to survive, to persist over at least some period of time over evolutionary scales. And like Evelyn Hutchinson said, the ecological stage is where we play the evolutionary play over the long term, and species interactions are the ones that determine who actually gets to play, and they shape the actors that play and transform at the same time the entire theater. So the nice example by Pablo shows how species not only use resources, they transform the environment that allow them to persist over time. And we teach our young students about these species interactions, like this is the Happy Things for 7th graders, which is a representation of an ecological network or food web. Because we know that if we want to say anything about processes in nature that actually are related to a given problem, like for instance, the effect of mass production of acorns in Europe on certain years, how much of that is going to help the cattle. We need to understand a little bit what's going on with the suite of interactions that happen around that system, even though it's overly simplified. Needless to say, if we want to say useful things about global change or about practical issues like fisheries production, we need to understand where and how the fabric of life actually takes place to support many of our human activities on this planet. How can we assess the importance of a given interaction between a species for the participant species and for the rest of this tangled bank? It's been an issue that, of course, preoccupied ecologists from the past 200 years, and it's been addressed by, I would say, two approaches, which are more complementary than disjunct, we hope. The first one has been the, I would say, simplifying or reductionistic approach, in which we select a subset of species which appear a priori to be strong interactors, as defined by MacArthur or Payne, species that appear to have strong effects, period, in a given system. We perform some laboratory experiments or field experiments or just collect new observations to quantify what we call the intensity of that interaction. And then we project results over space and time using either verbal models, conceptual models, or sometimes even numerical models that incorporate that information gained from a simplification of the system. The truth is that the tool has been quite powerful to examine the consequences of at least some interactions in the system. And in particular, I show you here two of the big gods of experimental ecology, and they happen to both work on intertidal marine coastal ecosystems in general, and it's pretty much where the use of this approach emerged into ecological sciences. Bob Payne and Joe Connell, unfortunately none of them is with us now, but they started this experimental way to approach questions related to species interactions. And just for those of you that might not be familiar with this, probably none of the mathematicians here, a classic experiment and a classic study in general in ecology is the Keystone Predator experiment performed by Bob Payne on the coast of Washington. And there is this mussel, the California mussel. Yes, the California mussel is in Washington too, and all the way to Alaska, just so you know. This California mussel is the prey of these sea stars. This is Pisaster lucretius, a beautiful large sea star. And below the limit of this mussel, there is a bunch of other species intermixed with bare rock. And if you add to this system Bob Payne and start removing this sea star from this area, you get what he called a monoculture. Basically, a dominant species expanded down and dominated the substrate, and by doing so, competitively excluded other species, reducing diversity. That's the classic keystone predation experiment that inspired, I would say, a few generations of marine ecologists around the world. Including Las Cruces. This is where we are at. This is, by the way, we didn't actually talk with Pablo about the slides, but I have a couple of slides that are the same. But this one is newer, Pablo. I'm going to share this slide with you. This is the actual labs where people can come and do sabbaticals, overlooking the ocean, solving those stochastic differential equations there. And Professor Juan Carlos Castilla, an emeritus professor in our department, conducted research in this very same area since the early 70s, mid 70s. And in the 80s, at the beginning of the 80s, he decided to perform this experiment, which was the exclusion of humans from that small stretch of shoreline. And that exclusion included mostly these guys, you know, fishers, but also tourists. And we've seen more recently the effect that also tourists have on coastal ecosystems. Well, these guys and fishers in general, fisher collectors, extract a number of species at different trophic levels, but one of the main species that they target is this muricid gastropod called concolepas, locally known as loco, that as Pablo said, is actually delicious. So it's the most, or one of the most prized marine invertebrates in the world. So there is an intensive fisheries conducted and pressure on those populations. And this is very briefly what happened after excluding humans from that stretch of shoreline, which created an increase or a release from predation pressure on the concolepas, the predator. And this is a study site within the marine reserve just few months after closure. This is about a year later, and you can see how halos start to appear in these, which is a mussel bed. These are not California mussels. These are tiny mussels that are not collected by humans. They're too small, called Peromidus, the chorito maico locally. And this is the way it looked by February 85, by 86, and it's about, well, it looked 10, 15 years after the closure. Basically, these guys, the predator had all but excluded completely the mussels from the mid-zone. So the simplifying approach to understand species interaction has undoubtedly provided great insights into the processes that regulate multi-species communities, especially when we try to understand local processes, and sometimes also explaining these visually very striking patterns of a spatial organization. But one of the problems remains with the approach that interests me, among many other things, including how generalizable are these things to different conditions and so on, is that in most of these experiments, we deal with a very small number of species. And in fact, the classic experiment by Bob Payne shows that there was a reduction from 15 to 8 species after the removal of the C-star, and he projected that those species were going to come down to probably just 1. The problem is that most ecosystems are composed by hundreds to thousands of species. More recently, one of my friends, John Whitman, conducted a review of experimental manipulations in marine ecology. He and his colleague reviewed 311 experiments published since the 60s, and shows several limitations inherent to the approach, including a spatial and temporal scale. But one of them is the fact that most experiments include very few species. In fact, usually the species that are manipulated are less than 10, rarely more than that. And like I said, most natural communities in all Earth ecosystems are composed by hundreds to thousands of co-occurring species. And that creates a huge problem when we're trying to predict consequences of natural extinctions, of species invasions, naturally occurring or by our own doing, or when we try to assess the impact of fisheries, like Isidora was trying to do with her network, because it's not simple as you might think to predict what's going to happen with the species right next to the one that are being exploited. And this was the basic uncertainty problem posed by Peter Jotsis in the 80s and in this nice paper in Tree in the 2000s. So how can we assess the importance of a given interaction between species in this tangled bank? The other approach to the simplification is, of course, the complex approach, I would say, which is how we embrace that complex network of interactions in the community. And the approach goes like identifying all the species that coexist in a given unspecified period, over a given unspecified period of time, over some area, identify the interactions that occur among all of these species. The species are considered as nodes in that network. Through the compilation of observations, usually diets in the case of consumption, or through experimental manipulations in the case of non-trophic interactions that I'll show you in a minute, and construct topological networks that predict or depict the species interactions. The first approach, complex approach to deal with the tangle bank and propagations of effects in natural systems was the food web approach. And in food webs, which are like the backbone of ecological networks, is a representation of who eats who, which species consume which species produce the energy necessary to maintain a given community or a given ecosystem, and how that biomass, which means energy and nutrients, is transferred to different species in the system. One of the problems we noted about a decade ago is that food web representation, even though it's quite complex, quite complicated as well, it's only a small representation of the actual interactions that take place in that tangle bank. In any real systems, species interact not only through transferring biomass, but also through other forms of interactions that we simply call non-trophic interactions. And this is nothing strange. You all have watched National Geographic and have heard of tons of examples of incredibly nice and complex non-trophic interactions. One of the simplest ones is habitat provision. A forest. You know, the forest provides habitat for many species that would not exist there without the forest. And that happens in all systems, like, for instance, mussel beds. The mussel beds are not only prey, they also provide refuge for a large number of species that inhabit under these. And in many cases, we get these really nice complex systems where a predator that eats primarily mussels is also benefited by its prey by reducing the risk of predation and ameliorating environmental conditions for the predator. This is a neat system that one of my students, I think is also in this workshop, Daniel Valencia, is trying to address both experimentally and also modeling this potentially very sensible interaction to environmental conditions, at which point this interaction is strictly a predator-prey type of interaction, at which point it becomes a predation, but mostly a facilitation type of interaction. But that not only happens in marine system, it's common to all ecosystems, from Finding Nemo movie, the associational defenses, pollination, trees providing nesting places for birds, and so on. We then try to provide a way to integrate trophic and non-trophic interactions with a bunch of friends and colleagues, and develop a sort of conceptual model on how to visualize a node that needs to acquire energy, process that energy to transfer the energy into production of new individuals. Some of that energy goes into respiration and excretion. And then the non-trophic interactions are those that either modify the rate of acquisition of biomass by other species, or directly modify the assimilation process once that biomass is being acquired. And there is a proposal on how to model this that is not too different, or actually the same basic model that Isidora is working with, except that we already included in this case an immigration and immigration process of biomass. So the model that we started with was already open in that sense, in comparison to the balanced biomass type of model that we've been working with Isidora. And with Pablo and Rolando, we proposed an stochastic version of that model that is what we've been trying to retake from the basic closed food web model to incorporating some of the ideas of an stochastic system that I think is very important as the next step to model real ecological networks. And I hope I have a little bit of time to tell you about that later. The other thing that we've done over the past few years is to provide at least one example of a real ecological network, meaning at least one where in a given area or a given stretch of space and time, we can say this is the way species interact in that system. Which species? Ideally, all species that we can count over that given area. And we've done so here in the same place. This is actually the same time that Pablo showed without the new buildings over here. This is the Marine Lab and this is the Marine Reserve. And we decided that it was one of the ideal places to construct an ecological network, because there is not only good information about the trophic interactions since the 70s, early 70s, but also a long history of experimental manipulations, which are needed in most cases to quantify these non-trophic effects. Unlike the trophic interactions, non-trophic type of effects are not binary. They don't occur or don't occur, but are strictly a function of abundance of the effector, of the one that produces the non-trophic effect. And this is the depiction of the food web of Las Cruces. Is the same one that Isidora showed early this morning in the first talk, just using a different representation of the same species. I think is Isidora would now know better. I think it's 116 species that are commonly found in Las Cruces at different levels, just to show you that they are heavily harvested at all trophic levels, just like Isidora was showing. So the artisanal fisheries is already a very complex problem. They exploit highly interactive species at all trophic levels in these communities. And this is the same network but now with the non-feeding positive interactions, which occur, they are not that frequent in the system, and the non-trophic negative interactions, which are quite common in this system. So this is the ecological network, and one of the few that has been documented. Is extremely limited in many ways, but it is kind of a glimpse into how a real ecological network would look like in a system. We also asked some basic question about like whether there is some structure to this tangle bank, to that ecological network. Do they all interactions of this, what we call multiplex networks, that has qualitatively three different types of interaction: consumption and positive non-trophic and negative non-trophic. Was there a structure on that? And the short answer is yes, there is a non-random structure to that web that I showed you before. And in fact, pairs of species interact mostly through one type of interaction, rarely through two, and even more rarely through three different types of interactions. And, but they can still be quite important for the rest of the community. And this fact that you have sets of species where you interact through trophic, non-trophic ways, it becomes really interesting to model, and that's what Daniel is trying to address experimentally. We also, with collaborators like what Miguel showed you, since we have this contraction, we could start exploring how trophic and non-trophic interactions change across environmental gradients. And he gave a nice talk showing some of our results. Modeling only the food web component of this network, since we believe they are pretty well resolved, particularly for intertidal, but also subtidal systems. Isidora, with our colleague Fernanda Valdovinos, have been modeling the impact of fisheries and what consequences fisheries may have on other species that are not exploited, and how that varies with variability in environmental conditions or productivity, which is what she's done recently. So that's pretty much all what I had to tell you directly about what we've been doing, and I thought it might be good to leave you with some questions, only if I have time. I don't know how I'm doing with time, Leonardo.
You have time, yes.
I have time.
Okay, I'll go with basically what I like to pose for the future seminars from the point of view of the people interested, or for people mostly interested in network, is how we can move forward from where we are at. From my position, where I'm sitting, is the empirical ecologist, where I looked at these things as approaches and tools to answer important questions that we could then use to address issues of sustainability of fisheries and so on. So the question is whether we are building an approach that can answer some of those practical questions, and whether that heuristic framework can really help us diagnose the state of ecosystems, or eventually, ideally, prognose some of the state of these systems and the consequences due to, for instance, that thing called climate change. And I see several limitations that need the combination of, I would think, theoretical work and empirical conceptual work, so that this framework can be pushed a little bit further into the practical real world. One of them is something that we talked with Rolando and Pablo for quite a while, and as you know, species in ecological systems not only exchange and modify biomass transfer among them, or they affect biomass assimilation, they also exchange information. And that is something that still has not been included in real ecological networks, and in general is still just starting to be included as a theoretical approximation in networks theory. Species clearly have memory, and ecological systems definitely acquire or lose resilience as they evolved. So basically the same climate disturbance that creates a given impact or doesn't create a given impact today can create or have an impact in the near future, because the state of the system has acquired some memory to previous perturbations. Similarly, the loss or addition of a given node, a given species, must lead to some rewiring in the system. And that rewiring of links is probably highly variable and probably also highly stochastic. But we have no idea which are the rules in real communities to rewire a given system once you lose them from there. What we are left to assume is that the removal or addition is a little bit arbitrary, and we assume that simply we lose those links. In terms of the ATN model, the allometric trophic network models, that like the ones that showed Isidora, those have become increasingly popular, and because information is more and more available about entire food webs and about body sizes of the species. And that has the beauty that knowing body sizes, you get an idea of metabolic rates, just like Pablo showed. And in that sense, it seems like you can parameterize a given model of food web and understand dynamics and even project under different scenarios. Like already we mentioned before, to me, one of the main limitations of the approach is that it's really unclear what the ecosystem level carrying capacity really means. I am not sure what it means once we fix it at a given point, and related to that, it is not unclear what is the food web to which the biomass balance implicit in this ATN model actually applies. Where is that food web where I should observe a mass balance? It is unclear to what extent also this allometric energy-based approximation really represents useful approximations to interaction strength in those ecosystems, or for instance, to what extent non-trophic interactions completely alter or not that energy-based species interaction strength. The non-trophic interactions now, the ones that I was just showing you, we provide some, I would say still a cartoon representation on how to model them. Sonia Kefi has been working on some approximation, but to me it is still quite unclear whether the time scale implicit in modeling food webs is commensurate with the time scale of trophic or non-trophic interactions. Some of them to me might necessarily move at very different time scales. So for instance, one of the problems is that non-trophic interactions, like I said, are inherently density dependent in very non-linear ways, meaning oftentimes they are a threshold type of function. And the typical saying, no, a few trees don't make a forest. You need at least a bunch of those trees of the same species to become a real forest. And when that happens, to me, it is not often clear whether we are modeling the interactions under the forest, whether we are modeling the interactions in the prairie, or somewhere in between, which really doesn't exist. So there is a problem on how we approach these non-trophic interactions and make them comparable to the consumption that are much more well developed. Many non-trophic interactions, like escape responses by prey that are exhibited not only by mobile prey, also by plants and everything, they always or most of the time have a very significant response to predation. Those responses are necessarily plastic. If not, they would become fixed in the population. And since they are plastic, they must vary over space and time, or there would not be a response like that. And it is not clear whether modeling the mean field, the average condition for a given prey species, really captures dynamics of a given food web. And I think that the approximations that we are using might be missing some inherent biology of how non-trophic interactions occur in nature. Most attributes of ecological networks are dependent on completeness. And this is related to the same issue of whether we can or not use the balanced closed biomass, closed system approach to model ecological networks. It is really impossible to include all species in any natural ecosystem. It's absolutely impossible, even the one, at least today. Even the one in Las Cruces doesn't include those things that almost nobody cares, the scam on the rocks, that scam on the rocks that Clara Arboleda, I think she was still around, actually have more species, more different OTUs or ASVs or whatever than all the species I've been showing you for Las Cruces. Different realms are in that scam. Those are not connected, not included in the network I show you. So there is no way to always, I think, include all the species, even the networks within lakes. The most complete networks within lakes do not consider the fact that pretty much all the species of insects fly and connect with other things in the terrestrial, in the forest around the lakes. So the question is, how many species must be included to say something about a given topological network, about a given network dynamics? And that is not clear to me. Since the number of species increases with area, where do we stop? Which is the subject of our model? And I think that finally, I think that one of the things that we should still work on is that networks are static representations of species interactions, and the same issue of completeness. We want to represent all the interaction that a species has with others in the system, but that occurs over some unspecified period of time. It's undefined how long you have to see and observe a given system to determine that you capture all the links in the system. And that creates the problem that we do not know what is the time scale that a given network really represents. Are we within the ecological dynamics that we want to model? Is this over evolutionary time? And that is a conceptual issue that for which I don't have a very good answer, but it bothers me that we are not thinking on how to define those interactions in a way that represents what we want to model about them. It is clear that over a period of time, species do not interact with all the species in the system. Probably within ecological time, those species will not interact with all the ones that we represent in a given network, and that for sure will have consequences over most time scales that we want to address when we move to practical issues, fisheries, conservation, marine reserves. The longer we examine individuals of a species, the more interactions are revealed. So how long should we observe those species? And many species change widely in diet through ontogeny. That variation means that many of the links that we represent in a node do not occur simultaneously with the rest of the links in the same node. They are not contemporary, and that for sure will have impact in the dynamics of that network. So lots to do, basically. That's the final message, that the positive things, looking at the glass half full, is that there is an open field for things to do, at least if we want to push or move forward the network theory to address more practical issues, I think. So that's it. That's all I wanted to say, Rolando and Leonardo. And I think that now I'm way past the time.
Yeah. Thank you.
Thank you, Sergio.